I'm actually going to turn you guys to the right a little.
To the right.
Because my tracking has come back in, so the next move will be 30 meters, 330.
330. All right. Co-pilot, you know what to do. Looks great.
And in the
Bridge,
ROV's sonar, which if you want to flip to camera 3, you can see we are
30 meters,
nearing the wall.
330 degrees, decimal 2.
Both on the high pack, but also the sonar.
30 meters, bearing 330, speed decimal 2.
That's a good copy. Thank you, Bridge.
This track line, we
RV nav bridge, move initiated.
basically are heading west for
Roger, move initiated.
the start to finish, but with a couple dog legs southward. If we start climbing the wall and encounter a good rocky ledge, then we'll follow that laterally to look at the geology and the biota living on it. That's our hope.
Get the fish ahead on the right.
And we'll climb about 100-meter vertical section. I mean, we don't expect the slopes to necessarily be vertical. That would be nice, but vertical relief of about 100 meters, and then move across a terrace, and then up another smaller segment of this giant block of rock that detached somewhere between, Jason Chater estimates, 14 and 16,000 years ago. And it is referred to as the Currituck Landslide. And for those of you who are unfamiliar with those processes in the deep sea, these are submarine landslides, where massive amounts of material detach from the continental margin and slide downslope. And our dive today is looking at the wall that is at the base of this large chunk of rock that fell. And I saw an estimate, Jason, maybe you can give us an idea. I think your estimate said 180 cubic kilometers. Can you give an idea of what that might be for those of us who can't think in cubic kilometers? I tried to get another unit of measurement that made sense to me, but...
You want to go for it again?
Yep. Look for the head.
Yeah, go for it.
Yeah, we usually, when I discuss these with
nothing to do.
various schools, I usually pick the closest football stadium or something, and use that as a guide.
Yeah.
It's north.
Because I'm here, it's Gillette Stadium, which
straight ahead of D2 series.
D2.
From New England, but I usually
just
end
be
up
in
not in progress.
Should be right.
Actually, the block that we're going up is one of the only pieces that didn't move.
Something about that pile to the left that the
when the
urchins
slide happened. So everything around it kind of was displaced, and this part, for whatever reason, stayed, and actually gives us a picture of what used to be here.
Oh, I see.
And Jenna Hill, who was on the chat next door, she led a paper on discussing some of the morphology of this from some seismic we collected a few years back.
Yeah, copy that.
So, but yeah, 150 plus cubic kilometers, you think, actually
tilt down. Just want to
was
frame all these
displaced during the last major landsliding event here. There was a smaller one south of this block, which was about 5,000 years ago. But,
some sort of urchin.
Yeah, it's hard to visualize that much material.
Soft tissue.
There's no real easy way to do it.
Jelly?
So you can't give an estimate of how many football stadiums that is?
Yeah, it's, I'm doing the math.
Okay.
It's about 10,000 plus Gillette Stadium.
Wow.
Listening from New England.
And here it looks
Filling
like
it from the bottom to the top.
Wow, incredible.
here it looks like we have a feeding frenzy of urchins on a jellyfish that is on the seafloor. I was wondering what was attracting this many urchins to one spot.
So sit right on top of the, real close to it and tilt straight down with lights.
Another of the pseudoanthomastus on the right.
Video, you want to start to come in?
Yep. And maybe kick on the extra lights.
Go Pat, go for lights.
And Scott France pointed out that there have been a few recent studies that looked at the importance of jellyfish as food falls in the deep sea. I actually saw one yesterday also. It didn't have quite the attention that this one has.
Go ahead,
but
Bridge.
Just wanted to let you know that movie's just about.
Well, it was in a burrow, so I wonder if somebody had grabbed it and pulled it down.
Yeah, that could have been.
0.6. If now would be a good time, I would like to adjust.
But we know if it was that one fish that lives in burrows, he would have sucked the whole thing down in one gulp.
Yeah,
we had some very dramatic feeding yesterday.
And surprising.
Yeah. And startling to those of us looking at the monitors intently.
There's a lot of tiny amphipods, probably amphipods feeding on that as well.
Oh, okay. And this is Stephanie Bush.
Move is complete.
Copy that.
You're above my star.
Yeah, hi, I'm Stephanie. I'm from the Smithsonian Natural History Museum.
Turning back
and I just came in because I like food falls. And it started getting interesting. We saw, what, like 2 corals now? So it's like, ooh, no offense mud, but nah.
You just wait till we see rock.
Well, yeah, exactly.
I know, you even like rock now.
Looks like we're starting to see something on Sirius. It's not defined.
Oh, a Bersingid starfish. Sea star.
Can we take a peek?
Yeah, stand by.
Wow.
Wow.
Did anybody have an identity for these, like, red and white beautiful anemones?
No, you know, several of us have seen them before, but none of us remember a name for them.
Because we haven't seen those on previous dives, right?
No, this is the first time.
All right, you can kill lasers, go pilot.
And that might be a different urchin also. It's a little more pink.
Video, let's start to come in.
So what is the rocky or muddy stuff on the behind this rock?
Yeah, look at that.
Yeah.
Is that a former sponge? Maybe a Faria type sponge that's
It has a base that looks like it. Yeah.
Wow, what a beautiful scene.
It'd be great for somebody's aquarium. If you had
to keep it really cold.
Yeah.
4 degrees C
a
aquarium.
Chiller bigger than the tank.
I wonder if that round, well, there's actually one of the rock-dwelling holothurians.
The Solus.
Yeah, the Solus. And then I wonder that the round thing is up for that.
Tunicate.
Like colonial tunicate.
Yeah.
That's what I was thinking.
Extra lights.
Go for it.
Yes, and the water temperature is 3.5 degrees C.
Lose the lights.
ROV nav bridge.
And Bob Carney pointed out that
I'd like
you
to adjust her
can
head
tell from the video that there's just really not a lot of flow at all here. It's very calm. Yeah, it'd be interesting to see whether these two urchins are the same species but just a different color.
Is that a siphon we're looking at right just to the right of the rock? Oh, just pulled in. I couldn't tell if it was a siphon or a worm.
I didn't see it.
And there's a pteropod shell, Stephanie. Did you see it?
No.
Right in there.
Oh, wow.
Look at that. It's a big one.
Oh, yeah. Cleo pyramidata.
Video, it's time to move on.
That one supposedly has a worldwide distribution.
Okay, Violet.
All right.
Who would have thought we'd see so much biology on this flat sediment plain?
I'll go to 280.
Copy. How's that look?
Okay, right there, that's 280.
Copy that.
Tilting up.
280.
All right, here we go. Another 30 meters.
Our overnight mapping of
like
this area included backscatter, which is a measure of how much sound is returned to the receiver with each ping of acoustic energy that is sent from the ship. It measures water depth based on how quickly that sound returns, but also a measure of its intensity gives us an idea of how hard or relatively soft the material is. And
Bridge, ROV nav.
Clearly showed that this substrate, this mud substrate,
20 meters,
was not as hard as the wall that we are approaching, which we are hoping to find some good rock outcrops that will provide lots of suitable habitat, but also some good rock exposures that our geologist, Jason Chater, who's on the line, can help us identify the rocks. His interest is looking at this wall's geology to help understand better the Currituck landslide that occurred in this region about 14 to 16,000 years ago. And the U.S. Geological Survey has done a lot of research looking at these continental slopes of the continental margins to determine where areas have detached and volumes of material that have subsided or in a landslide type of setting. And here's some good rock debris.
Similar to the first one we did, but he seems bigger.
Is more concentrated than what we've seen. We've seen just a few pieces of this type of material farther out. See lots of the fringed sea stars.
Inch closer.
And this is another of the halosaurs that we saw earlier.
Copy. I think they ID'd it.
Yep. One more time with lasers.
Yeah.
And if my Latin serves me correctly, that would mean salt lizard.
All right.
Why are you looking at me like I know
There's
Latin?
something blue, like
You're a biologist. I thought all biologists spoke Latin fluently.
There's something blue right behind it.
Well, this is some cool rock, though, isn't it? It's
kind of a strange blue thing.
Jason, you want
to the right of your
talk about it?
Yeah, right.
Right there.
Yep.
A little bit up.
Is he talking to himself on mute again?
Oh no, Jason.
Plastic.
Unfortunately, that looks like a little marine debris right there.
It does. Thanks for looking, the pilot. Nav was wondering what that was as well.
Not a problem.
So this is heavily bored into by some marine organisms. Possibly those mollusks. Stephanie, are you familiar with? There's a mollusk that does boring into these clay sediments.
Yeah, I don't really know about those bivalves, especially not this deep. I usually think of like shipworms and stuff, but I don't know. I'm sure there's There was some
it's a big one back there.
Anthomastus / pseudo-Anthomastus
There we go.
amongst the Brisingeids.
Pioneers used to ride these babies for miles.
A big coral in between it.
Yeah.
It's a vertical
that way,
thing.
right?
We
On the
are.
rock to the left.
Awesome.
That was only a 20-meter move, and it should be just about coming to an end.
All right, I'm functional again.
Lots of time.
So these look like mudstones or clay stones. As you mentioned, they're pretty heavily bored.
Tracking looks
by,
like it's
leading
much more
to a lot of
accurate now, which is
bioerosion,
good.
which weakens the rock
The
to some
tracking
extent.
switch back to on.
They're moving up into the debris
Well, I'm
apron,
actually
or
wondering if the FRB
sometimes called a tower slope
work
at the base
is
of
causing vibrations or
the
something.
steeper.
RV9 bridge move complete.
So we're probably
Big
going to see a
bridge.
mix of different rock types
Move complete.
that
Copy.
form the sort of the bulk of this big intact block that didn't fail. So I expect to see a lot of different types and levels of
Did you want to
erosion
go for
that
this?
are affecting things.
Ah.
Because
See here, some of the pieces
Just going to
really
have you hang
tabular.
out here,
Good.
co-pilot, to see where we
They
settle
do
out.
some very good layering up top. And I'll stop tilting now, given
Look at
that
there's like white Eumenida or white squat lobsters in there. Oh, yeah, that one. Is this like a weird, like, net of it? You think it's bamboo that fell down?
It looks like a bunch of bamboo corals, doesn't it? Scott Francis, a bramble of bamboo corals. Yeah, and I've
Maybe
definitely seen
tumbled down from somewhere else, or do you think these were
Tumbleweed of bamboo.
I
these grew here?
Look at the left branches.
can't see what they're growing on, but there's
Left and up
some
from lasers.
some rocks and
up.
things, maybe small ones.
See all the branches.
There's also desmophyllum cup corals and white little squat lobsters that are possibly Munidopsis species.
There's a bunch of gooseneck barnacles growing on it, too.
Barnacles.
I think that's a separate one.
And
on
maybe,
the right.
yeah. Did you say cup corals?
Yeah, there's a
Sorry, yeah.
couple of
Yeah,
desmophyllum, which are the white solitary corals with the orange tentacles.
Gonna kill lasers.
Watch lead nav. I'd estimate we've gone a second 100 meters.
Watch, is anything in particular in
Very
this
top
bunch?
corner shows the root of this one.
Yep.
There's also a sponge and orange brittle stars, and
Go ahead, Bridge.
I saw something walking away on the lower right.
and Scott France said that this might be Currituck flexibilis, which is known to grow in a bramble shaped like this. I've never seen a
There we go.
bramble of octocorals. It's so interesting.
They're relying on themselves for substrate, I guess.
Tilt up or down.
I guess it's kind of
upside
like
down.
Lophelia does too. Providing their own substrate, growing on top of other colonies.
Although I couldn't see it clearly, it sort of seems like at least 4 thick sections, which may be 4 different of the same species. Did we get up with lasers yet?
Yeah, we had lasers on for a while.
Okay, there's an actual foothold at the very bottom left, just went off screen.
Bottom left. Oh yeah, let's go for that.
You can see a test from an urchin
Looks
there.
like a shark swimming on your starboard side.
Ooh.
Oh, and we might have
Small one.
Washley, do you want me to leave this?
Yep, go ahead.
Copy.
That's
Okay.
great.
You
It
see the
goes
base
tiny
of one
one
of
up on your
those
starboard
corals
side.
too, though.
Yeah,
Not sure how high.
Copy. Let's
we
see
had
some
a
of the shadow.
report of a shark swimming by. Just see if we can catch him.
got lasers on.
Not literally.
Well, not literally. Yeah. Just
We're
look
pretty
at
sure we can't. There's a little sea star, our first sea star of the day.
Wow, look at all those Brisingid sea stars.
Oh, that's right. Of course, we've seen
like
Brisingid.
Oh, maybe this is another octocoral, Paramaricia,
yeah.
maybe.
Oh, is that what that?
I think the shark swam off. So.
Okay, well, too bad we missed him.
Too far.
Maybe he'll come back, or maybe we'll catch him again.
Is that what that orange or yellow coral was earlier?
Yeah.
Okay.
Well, this sure was an exciting group of rocks.
Leslie would say all groups of rocks are exciting.
I'm just going to be
true.
quiet. I know you
So
realize
I'm
that.
saying it for you.
Okay, thank you.
I rotated a few degrees to get into view. Pilot.
Copy that.
Pseudo-anthomastus slash
point,
anthomastus.
Correct, the red
Square up
octocorals.
to the slope and proceed that direction.
Copy that. I'm going to get a quick zoom on Sea Star. And
Whatever
then
works best for you, co-pilot.
Copy.
video would come for Sea Star.
Sea star and possibly another, Paramaricia on the right with another one of the snake-like ophiuroids.
Do you want lights video?
I don't think we've seen
No,
this
those
one.
lights will shine underneath of this.
No.
Yeah, this
In
one
our
doesn't look
travels?
familiar. This looks like the one that has like the big
More like we need lower lights.
portal on the dorsal side, which I know that's not the right name for it, but
So you want me to bring down the lower?
I wonder if
lower
Chris
signal?
Ma is up yet this Sunday morning,
I don't know if we're gonna
or if
sit
he's
here
still
long enough for it.
dreaming of
Yeah.
echinoderms.
Do the same thing. Maybe just get a couple good views and
and a bunch of
have what we got.
polychaete feather duster type worms on the left.
Yeah, it looks like a wall in a haunted mansion full of spider webs, actually.
At least the TV version.
Right, yes.
Yep.
The possible shark.
Forward copy.
You can see it in your starboard rail. moving
Pilot, we'd
towards
like
the
to
front.
collect this rock, please.
I think this one just breaches our upper
Just
limit.
Time for a new rock box.
It would make everyone so happy, though.
That might be time for a new winch.
Maybe we could have a trailer on the back of D2.
Not sure the ship could carry that one back.
He's kind of off in the shadow somewhere up there.
Huh.
Yeah. In the shadows, upper left somewhere.
I think the potential shark might be lurking around here somewhere, Washlights.
No, I think he.
If you want
swam
to
up.
take a scan, that's great.
He was heading upslope.
Upslope. Copy. Maybe we'll catch him later then.
Yep.
You know, Nav, if you really want to get a pilot to go,
See another
really
of
get into
those
detail, it's
anemones
octopus
on the
up
base of
towards
that rock
upslope.
on the left.
Don't see him.
Maybe Actineris is the genus. It might have a sister
Co-pilot
species
Nav.
in the
Either
Pacific.
of you want bottom time.
The anemone? Is that right?
Just past the halfway point.
Actineris.
Can we take a quick zoom where the lasers are, just to the left of the lasers now, on the seabed?
Yeah. Video, go for quick zoom.
Have you piloted?
Wow.
Since that last time that you piloted when
Do
I
you want
was
to get closer, Washlights? Or is that good?
No, that's okay. It's
got
good.
it?
I don't think so.
This is
go ahead and pilot.
Sure
a
you don't want this
remnant
last one,
of
Fernando?
an
I piloted
octocoral,
yesterday and the day
and
before.
it's
Alright.
totally covered now with brittle starfish.
Is that just Brisingins too? They look white.
Yeah. it
Oh,
looks like
the thing that Leslie was just pointing to.
Yeah,
Just
do we
lower left.
want to look at that?
Lower left.
Sure.
Yeah, it just went out of the picture. It's at the base of this
Bottom of the screen.
Spongy?
boulder. Yeah, right, the white.
Copy that.
looks like a sponge.
Cauliflower sponge.
Look at all the
Me
Brisingid.
making up
They're
common names
beautiful.
now.
Say, I've never heard that.
Alright, video, go for it.
This might be like a
Kill
Faria
lasers.
type sponge, glass sponge.
Get all the tube worms. They're out. That we
Is that
don't
a
seem to be
carnivorous
disturbing them.
sponge
Korea?
there in the front of
What was the Pheronoma? Pheronoma? I'm just adding things together and making up my own names now. Great.
Look at the branch one at the bottom.
And what is that? Is that one of the little wishbone thing? That's what we were seeing the other day.
Yeah, we didn't know what those were.
I think they're sponges.
Yeah,
I think it's another kind of hexactinellid.
Well, that might.
Those were. I thought they were carnivorous
carnivorous
sponges.
ones. I think that those
So
are the
another
carnivorous.
one at the top.
Yeah, there's one on the left and one that's wishbony.
Tilt up.
I think that's the carnivorous sponge.
Okay.
A different one
There's
than we
another
collected
one.
previously.
No,
yeah.
Are we good here?
Yeah.
All right.
All right, video, you can come live.
Are these
Desmophyllum, it looks like, cup corals.
cup corals? Yeah. We just switched our nav and pilot. That's a beautiful scene.
It sure is. The water clarity is so beautiful.
It's so nice to see clear water again.
Yeah.
Or to not see the water.
So many Brisingid sea stars.
Our video, let's go in the dark object just from here.
Nice, another tube anemone.
Is that full? Just want to get
Pretty
an
color.
ID.
We haven't seen one this color.
No. Is it true for most of these tube anemones that they have stinging tentacles as well as sticky tentacles? Or was that just the one we looked? Okay.
Yeah,
All right, let's move on.
they all have
Purple
both of those. I guess
inside and
Tina was making
octopus
it sound as if
effectively.
some species
Fossil
have
sponge.
more
Sponge.
stinging cells and or more potent stinging cells.
Yeah,
Are we
it's a
full wide?
beautiful crop of Anthomastus.
All right, let's continue on.
Okay, do you want me to center?
And our slope begins to increase.
Maybe the slope
You can
is
see how all these
this
rocks are detached from
It's hard to say.
someplace
I'll
upslope.
turn right, kind of look into the slope here.
copy.
Yeah, looks pretty good.
Maybe somewhere around here.
290.
290.
Looks good on sonar.
Okay, 290.
That's snap on the yellow rubble. Is that the edge of the rock? Yes. Okay.
A few more Anthomastus, the red
purple.
octocorals that you see, and a different sponge on the
Can snap on
edge of the.
purple.
Huh. What is that?
I'll look at it on the upper right.
Somebody ate an urchin, and that's like the little pieces of the test.
Maybe.
Looks a little bit like it. Like the test got crunched. I don't know.
There's a big pile of them.
Yeah,
in the
more of it.
top of the screen.
Oops, sorry, video.
Not a color you see out here every day.
No.
It does look good.
It looks like a quahog interior too. But yeah, that looks like an urchin. Is that what you were saying?
Yeah.
Yeah, that's definitely urchin right there, isn't it? The spines. Pretty color. That color you see in the bivalves that I call quahogs.
Video's clear.
Pilot's clear.
Isn't that like Pacific Northwest?
Pat,
Oh wait, no,
20
that's
meter
like
bout towards slope.
New England.
New England, yeah.
Yeah, sorry.
Oh yeah,
Would
right
it be
above
possible to get a little zoom on, that stalked organism?
Pretty.
Wow.
Ooh.
Is that a hydroid of some type?
I think it is.
It's awfully big for a
Doesn't it
hydroid.
look like an anemone of some sort?
You want lights video?
With tentacles?
We're too far away for lights.
Waiting for someone in the chat room to have an
Just come
idea
out a little.
for us.
Right there.
Possibly.
Just frame it.
And we have a bit of a
Okay,
delay
let's go in
from
on the
how
top.
quickly we see it versus when people tuning in from shore see things.
So delicate.
Yeah, it's beautiful.
I've seen pictures of something like
Yep.
this before, but I
Try to get
can't remember
closer with
what
the
it is.
extra light.
Megan
What are
McCullers says it looks like a
I don't
giant
think we
hydroid.
really can with
Really?
its position
Okay.
on the rock.
Wow.
About
Chorymorphid
Huh?
solitary
You can get above
hydroid.
it.
Wow,
Light
that's
shoot
beautiful.
Yeah, okay. Just come wide.
It's huge.
That's really pretty.
I'm going to go back in for a better look. Watch leads, so stand by 1 second.
Roger that, thanks.
Take a look at the rock with me. Okay, let's get fog lights on.
Isn't that a little bit too low for those lights? Oh,
Nope.
no. That's pretty good.
Stand by video.
Sorry.
All right, it's yours.
And Scott France said that the grape-like structures in the center are their reproductive organs. This is such an unusual creature.
Wow.
Nice job, video.
The
That
mouth in
is
the center there.
beautiful.
It is really gorgeous.
Tilting down.
Such a delicate looking.
Too much.
That's good.
I don't
creature.
think it would survive in a high current, high velocity current.
No.
Gonna try to. There we go.
And Megan McCullers says it looks like a giant
Just off in the
Tubularia
thirds.
polyp.
Full specimen.
Yep.
There's some more sponge, I believe, to the, and also to the right at the base of this rock. Saw
Yep,
some
left,
growth.
left
Video's
and
clear.
right.
Okay, pilot's clear.
All right, Pat's ready for
Can we
that
take
20
a
meters?
quick look at
Let's do it.
the,
Great.
where is it? Yeah, just that sponge that's sticking out at the base. The whitish material that's sticking out at the base of where you just were.
Go back in.
Sorry. Yeah.
Okay, ready for move?
Yeah.
20 meters at 290 degrees, decimal 2 knots.
20 meters at 290, speed decimal 2.
Good copy.
We haven't had a spongy dive in a while either. We haven't seen hardly any sponges recently, the last couple dives.
Is that good, watch lead?
Yeah,
Think
that's
so. It's
great. Thank you.
All right.
got a little sediment cover, so can't
Moving
really
on.
see a good view of it.
What was the bearing on
Wow,
that?
that urchin test
290.
is in shambles there. It's
Video, let's snap
Bright color.
here on this
Pardon
kind
me,
of
I've got
strange
pictures.
colored. Yeah.
Oh yeah, there's something brightly colored here.
got one of those sponges.
Is that the blue
Wow.
thing? that
Tunicate.
has the other
Pretty.
stuff?
Yeah, it
Around
does
some
look like
different.
a colonial tunicate.
Sorry.
What's on those
Coming
rocks,
around.
Leslie?
We can go back in
We have
in a moment.
a crust,
Just don't want to
precipitates.
get what we were just looking at.
Iron manganese.
Well, I'm not sure if it would be what the composition would be. It certainly has iron in it. Yeah, I've been saying iron manganese for most of the dive, but really only the elemental analysis will reveal what the content is
Copilot.
of the composition. Certainly there's iron.
Okay. Go ahead, video.
And a worm. Some worms.
Mm-hmm.
Our
pretty.
favorites.
That's a tunicate?
Yeah.
Colonial tunicate. Wow.
Actually, it could be a sponge. I'm
Oh
not
yeah,
sure.
actually, I think it maybe
could
is.
be a sponge. And we have been seeing an encrusting sponge that's like this in other dives.
So throw those extra lights on here, see what
Yeah,
it does.
it looks, on close-up, looks fuzzy like a sponge, not smooth like a tunicate.
And do we have,
Ooh.
what are these worms?
The bioturbators of these rocks.
Oh, supunculids. That's right, says Bob Carney.
Clear.
All right, let's see if we can cover a little ground before I get distracted again.
By a big shark.
Those are very good distractions, Pilot.
What are you talking about, distracted?
We enjoy them.
Well,
Or at least us biologists do.
not really distracted. I just said a couple times, like, all right, let's make some way, and then would find another brightly colored object, which is great. I'd rather be stopping.
Means there's something to stop for.
Exactly.
I don't know if we've ever, on this expedition, been able to see peanut worms or these little sipunculids that were
Pink
around.
coral, I think, is new.
Ooh, yeah, maybe a Chrysogorgia.
Wow, look at this over here.
It's a fin.
It's this satellite dish sponge.
You think?
I
Over
don't
on
know,
the
I'm
right.
just saying that. I don't remember what depth we got that from. That
It
was so long ago.
Go ahead, video.
We have seen so many things. It's just, yeah, it looks like a Chrysogorgia.
Slow scan down.
They're so delicate looking. Like,
Yeah,
so, like, such a thin skeleton.
it looks like they have an anemone and maybe a scale worm. It's a polychaete worm that you see the reddish thing towards the center.
1704 or jellyfish stuck there in the upper right.
Drifting on you as I turn.
Translucent to the right of the worm.
Yeah, I was trying to figure that out, Roland. But you're right, it looks like a Medusa that got stuck on there.
Oh yeah,
Full specimen.
it's amazing what we can see with this camera.
Lasers. Video's clear.
Oh, it's clear. Looks like another one of those little blue
Several.
spheres. Yeah, a couple of them in the area.
That one straight ahead left, maybe
Yeah, we
up
want
coral.
to take a quick look at that sponge.
That's a sponge also. Thought I saw polyps.
It sounds like
Let's go for
Scott
sponge
France
as we
and Les
come around.
Watling were having a hard time
Partial.
figuring out which Chrysogorgia species that was.
Wow,
And that's a beautiful sponge.
that is beautiful.
Nice.
Another blue one in the lower left.
Mm-hmm.
Did you want a closer video?
I think you're doing a real good pirouette. I didn't want to interrupt it.
My right toe's going to hit momentarily. So why don't you go ahead and we'll get both of them from this angle. Nice.
ROV nav bridge.
Go ahead.
Bridge, go ahead.
That move is complete, and the current is now from a 345 at 0.8. If it would be a good time, I guess I'll just keep
Roger.
chasing it. Maybe another 10 degrees to starboard.
Video, are you clear?
Yeah, this is a good timing. Go for it.
Okay, thank you.
Are we stopped, navigator?
Yeah, ship just stopped, and they're going to do a heading change.
Video, let's go
Probably
here.
10 degrees.
Real quick, get this tube worm before I leave. And see if that's
Oh,
also
wow.
the same Chrysogorgia above.
Oh, that's a nice,
It's a
that's
beautiful
nice
tube
tube worm.
worm.
That's really
Wow.
cool.
Yeah, and there's another Chrysogorgia in the background, maybe.
Coming down for 2. There we go.
Gorgeous.
Wow.
And when you're clear.
Slight rotate right.
Ah, you do have more. Great.
Just waiting for it to center up.
Yep.
And he's out for Sunday brunch, trying to find some food in the water column here.
Let's see if that's the same.
Huh, looks like bigger polyps on that one.
Yeah, this looks different than the other one.
Scott's asking if there's an egg case in the background. He might be talking.
Is that full?
That is.
Acanella? This one?
Ah, no. Well,
or is he referring to the?
Go around the corner.
Do we need a closer look at this one, watch leads, before we go back and sample the other
I
one?
think we have an idea on the one that we're just looking at, and it's Acanella, which is a bamboo coral and different than the one that we were
Understood.
looking at.
We're heading back to the
and we
other
also,
one then.
before you head back, though, we've had a request for a rock sample from Jason Chater. So we want to zoom in, he said, to the left of the tube worm. I assume he means the one to the right of the sponge. So let's zoom in and he can verify which one he's talking about. Looks like there's a fresh one.
I'm assuming this one right here on top.
It's looking like that, yeah, because it also has some of, yeah, that's a good one.
All right, let's come wide. We're going to go for the coral first, and we'll come back for this rock.
That's the one, Jason?
Yep.
Okay, great.
It's not in place, but it might be a good representative of the various processes.
You can see how pieces have broken off that larger boulder that's now moving into the upper right, too.
Jason, thanks for picking such a great site.
I apparently have a talent.
You could get a job being a geologist.
And not just play one on Deep Sea TV. Deep Sea TV.
I play a good one on TV.
Yep.
Yes, you do.
I think this is probably a good angle. Maybe I'll square up a little.
The layers in this
I was just going to
really
say,
cool.
you can
and
see
like
how they
perpendicular to where they, how they've layered.
This rock was
on the setup. Stand by.
Formed in place in layer by layer at a time, and then it's broken off and tumbled down, and it's fracturing along those bedding planes. And we've seen actually those larger blocks appear to be broken along those larger bedding planes too. So.
All right, co-pilot, we spun up.
Yep, we're ready.
Okay. Arm coming live. Numbers live. imagine just the a snip from the top watch leads.
Yeah, there is also a worm that's partway down, but
okay.
I don't want to take too much of the coral either.
Let's put fog lights on and go in on video, please. Does that help with the fog lights, or does that make it worse video?
Colors aren't right, but if it helps you see down below, I think it's good.
Just trying to pick out the worm, but I can't really see it from this side.
Yeah, I think it was about halfway down.
Okay.
See it right on.
But if we want to take less, that's okay too.
Just lower than half.
You direct me, watch lead, how much you want to take. All right, you can come partial.
You see the worm?
Yeah.
Okay.
We can probably take fog lights out. Just come out a little bit more. Okay.
This guy's super small.
Oh gosh, it's so small. I thought it was bigger than that. How many times have I said that?
You'd think we'd get used to it.
Yeah, I mean, I guess if we could take the top half of it, that's probably, maybe we'd get the worm, and there's enough of it to identify then, I hope. You can see the cutting blade.
Yeah, that's a really good view of it.
That they use to just take a snip of these corals, so then the rest of them can continue to grow.
Does that look like a good place to cut?
Actually, a little bit lower. Scott France is asking for a little bit more, so if we could go a little bit lower. Awesome, that looks great. Thanks.
All right, let's come on. I don't think you're going to get the box from here. Or what do you think, Kabal? Think you can get out?
Yeah, I think I'll hit right here on this toe.
I'll stand by here. I'll pull off some.
And for anybody who is watching from home, we don't make many collections on these dives, but when there is a question about the identity of a species, that's one of the reasons we would collect. So we had several experts on octocoral taxonomy in the chat room here, and they could not put a species name on this Chrysogorgia, so that's why we're making this collection. And when we get the collection up to the whole ROV back on deck, and we get to take the samples out, we'll preserve some of the samples for molecular work, for genetics, and that can also help with identifications. And then we also preserve the rest of the specimen for traditional morphological analysis. And so we have four boxes on the ROV, bio boxes that are meant for biological samples, and they keep the water nice and cold in there so that it doesn't get exchanged with the surface waters on the way up, which are much warmer and which will definitely hurt the corals.
I'm actually going to come up. I'm going to go in.
And then we have two rock boxes on the ROV as well.
I'm worried about this being a little bit lighter
and those
not being able to get it down deep enough.
Those don't need to be closed to the environment, so they have water flow on the way.
Opening jaw.
Up and down, or up, I guess.
Clear.
And that sample went into the port outer bio box.
Starboard inner.
Oh, sorry.
She was talking. We switched.
Okay.
And that is biosample 1. For those annotating.
Okay, leave hydraulics there.
Nice job, Pat.
I got that one annotated. since I'm here.
Yeah, a little bit of a last-minute change there on the box. I was worried that it was a little bit floaty and wanted to get it deeper down into the box, so I moved it over one.
Okay, that's great. Thanks.
Okay, over to the rock now.
Yep.
Looks like there might be another one of the Acanella, which is the octocoral in the lasers right now, over to the left. And for a reminder, the lasers are 10 centimeters apart. We use those for sizing rocks and fauna.
Okay, arm coming live, or arm coming out of index.
ROV nav bridge.
So,
Go, Bridge.
watch leads.
Yes.
Now's a good time.
Originally, we kind of pointed to
10 degrees more to starboard? Yeah,
Yes.
that's a good time.
a
Right.
rock
Right.
that the lasers are on. Is there more interest in the one above the lasers?
Yeah, it's a little bigger because I'm just finding out from Jason. It seems to be the same type, and it's a good fresh exposure. Could you just zoom in on it? And
Okay.
He's pointing at it.
yeah.
Go ahead. Let's go in video.
And Jason said, yep.
Okay. Go for it. That's nice. Definitely a good fresh surface.
You want to
And
try
then
with
the
fog lights?
Outer
video?
areas have
we're
some
good.
All right.
biological
on.
activity, and Chuck Messing noted that agglutinated forams appear to be those little branched organisms holding on to the older exposed surface. Foraminifera
let's
are
move that
single-celled
rock box in so I can
organisms
open it.
that particular group use whatever sediment is available to build their chambers, their chamber walls. And they live in the outer chambers, but the actual structure can be quite large, as these are indicating. Most of the foraminiferal species are much, much smaller. But these agglutinated forms can reach, a centimeter in length or more.
Okay. Ooh, it's quite soft,
Mushy.
actually. Do you still want it?
I'll wait to see.
You want to zoom in and
Hear
show
from
that again?
Jason.
Oh, you want to show that from a zoom in again?
No, I think we're okay.
Jason, what do you think? We can get it, probably.
What's your grip force?
Yeah, I'm watching to see squeeze goes to it.
Does he want us to try to squeeze all the way through?
Do you want to just keep squeezing, Jason?
Or piece?
Yeah, keep squeezing. If it breaks,
Okay, let's go in
we
partial.
learn something without having to bring it back.
Okay. We'll watch it.
Tilt up, co-pilot?
You want to maybe squeeze a different rock?
No, thank you. So I'm just going to go for the end here.
Very fine grain.
It's squeezable.
It's very.
It will not make a good thin section.
I will not.
But if you can grab a little bit of it, we can you can still get, information from it. But,
well, then it occupies one of the boxes, so we wouldn't be able to.
I don't think that's going to survive in the rock box.
Yeah, and we can't put it in a bio box.
It's compartmental.
If you could squeeze that remaining piece and break it, and then we could just take a closer look at it with the video.
Trying to keep the shadow out.
Kick on the extra light to kill the shadow.
All right, let's do that.
Kind of doing some field geology.
Yeah. No rock hammer required.
Before you move it, is that a fossil in there? Or is that just the grooves from the jaws?
Yeah, it's...
You want to slowly look at the backside?
I was trying to turn it around, but my jaw was...
Do we have a scale of squeezability?
That's a nice view of the forams.
It is very nice.
Are those the
if those are forams.
There's
they
a
just
fresh
took a jump.
piece.
A leap of faith.
Yeah, that's it. Can you rotate that a little bit so we can...
We'll try. We'll see if it... Hopefully it doesn't shift on us.
Why would parts be red like that?
Iron?
Iron?
Yeah, I'm wondering if there were sort of micro fractures on it from the tumbling it did, and maybe some... It could be bacterial or something, too. Microbial growth. All right.
Yeah, that works for me.
All
Thanks for trying.
Yep.
Pilot, this is Watch One.
Go ahead.
Before we leave this area, could we zoom on one of the red anthomastus?
Yeah, who would have thought those would be so friable?
I believe.
Yep.
Thanks very much.
Makes you wonder how they tumbled downhill.
I know.
Quickly being destroyed.
Maybe they didn't tumble very far.
Yeah, everything around here looks pretty soft.
And I imagine that that's why there aren't more octocorals and other corals growing on it if it's that soft.
Yep. Even the main rock is quite soft.
Interesting.
And why that bamboo coral that kind of is the tumbleweed is like one of the biggest things we see. It's like making its own structure, not dependent on the rock.
Thank you, pilot.
Fog lights off, please.
Oh, it doesn't look like the lid.
Oh.
I think we ever put in those new springs to prevent.
Do I need to go?
From closing all the way now.
Okay.
Let's bring that back in.
It used to close itself, right?
It used to.
So our up close and
springs, and
personal view of that fresh surface.
Spring